Hi friends, welcome back to my channel Only Bio, presenting up the revision of the chapter Biotechnology. Friends, as you all know, biotechnology is a very important and a very vast chapter. So I have divided it into many parts, and this is the first part of this chapter, which includes the introduction about biotechnology. So please follow this series, and as we go ahead, it's going to include all the complex points in it, like maybe gene cloning, plant tissue culture, the fingerprinting technique, and so on. So please keep a check on the playlist as well. And as been demanded by the viewers, I've also recorded it in Hindi. So for all those friends of yours who want the same chapter to be explained in Hindi, so I have attempted the same. So please, you can even share this video with them so that I get to know. And also, I am waiting eagerly for your comments and your suggestions, your likes and your shares as well. So, let's begin with. To start with, what exactly do we mean by biotechnology? How do I define biotechnology? An international committee on biotechnology in the UK has defined biotechnology as the application of scientific and engineering principles to the processing of the materials by biological agents to produce goods and services. Friends, biotechnology, we can call it as a multidisciplinary science because it's a fusion of different branches of science. Like we have engineering, we have technology based branches, we have microbiology, we have biochemistry, we have genetics, and all these being merged together. And this has all been done for the change to be brought about in certain materials with the use of biological agents. Friends, biological agents here mainly include the microbes like bacteria or yeast or the enzymes produced by them, either to bring about a change in the substance or give a best service. Next, the basis of biotechnology mainly consists of engineering and different sciences like microbiology, genetics, and biochemistry. As I did tell you, it's a multidisciplinary science, so it includes all these branches and all these being merged together, and then comes up biotechnology. Today we know many more areas of biotechnology. Friends, like information technology, biotechnology is also a very challenging field, and it includes many more areas like immobilization of cells, antibiotics, monoclonal antibodies, vaccines, production of vitamins, organic acids, and so on. Now, what does it mean? Friends, we know that either we use the microbes or the enzymes that are produced by them. Right. Now, many of the times the enzymes are intracellular. Friends, I hope you know that the enzymes could be either extracellular, that is, they are released outside the cell. So, we need to just extract that enzyme and bring about that process. And many of the times, the enzymes are intracellular. That is, these enzymes are produced within the cell, but they are never released outside the cell. Now, if a particular process is dependent on such intracellular enzymes, we need to spend a big money on isolating these intracellular enzymes, which becomes really very costly. So, if we want to cut down the cost, the scientists thought over why not separate the cells and use the cells as a whole and bring about that process, because that will be very cost effective. So, that is what is meant by immobilization of cells, to put it in short. Second is antibiotics. Friends, anti is against, biotic is life. These are the substances that are produced by certain bacteria against another bacteria. So these can be readily used in protecting ourselves against disease causing pathogens. Then comes monoclonal antibodies. Friends, antibodies are the protective chemicals, proteins that are produced in our body as a response to the attack by foreign organisms, that we call them as antigens. So sometimes when the immunity is definitely compromised, if the person is immunologically quite weaker, scientists thought of why not increase the production of antibodies by this particular method, wherein we use a particular cell which is sensitized against the antigen, fuse it with a myeloma, that is a cancerous cell, so that all the antibodies that are produced are identical. And by the use of the same, we can certainly help the patient out of a particular disease. Then are vaccines. Vaccines are the protective substances, might be the live microbes, yeah, certain products by means of which we actually sensitize the immune system so that they get ready by producing the antibodies. Then is production of vitamins and organic acids, which are really very important for the body. Next, let's understand the concept of gene. The transmission of characters from parents to offsprings constitutes heredity, and its study is carried out in the science of genetics. Please remember, genetics is not only the study of similarities, but it also is the study of variations, and all this collectively is heredity, that is the science of genetics. Gregor Johann Mendel in the 19th century made genetics into an experimental science. He crossed different varieties of garden pea, that is Pisum sativum, having different characters, and observed how traits are expressed in subsequent generations following self-pollination. Friends, Gregor was a title given to him. Johann Mendel is a name, and he was the first scientist who gave and approved an experimental evidence for genetics. He had crossed up different varieties of garden pea, which is scientifically called as Pisum sativa, which has different characters, and he observed how traits, that is, characters that are expressed in subsequent generations following the self pollination process. He observed that there was something in the plant germ cell which controlled the transmission of characters. Mendel called these as factors or units. Friends, remember they were also called as determiners or particles, and that is the reason why the concept was called as the particulate theory of inheritance. On the screen, you can see the great Gregor Johann Mendel, the father of genetics, and slightly towards the left hand side of the picture, you can see the Pisum sativum plant, that is garden pea. These factors were present in the gametes, that is pollens and egg. These gametes form the connecting bridge between the parents and the offsprings, that is next generation. In post-Mendelian period, the researchers were mainly interested in knowing the nature of genetic material, that is of a gene. Friends, post-Mendelian period, it includes the period after Mendel. So the researchers, they got curious about knowing the particles or the factors, which Mendel had called them as factors, and later on the term given for the same was gene. Let's understand the historical account of the same. Let us have a bird's eye view on the historical account of the discoveries associated with genes or nucleic acids. Hofmeister in 1848 for the first time observed chromatin threads during cell division. Friends, cell division, if the cell is not dividing, what we see is the chromatin network. But if the same cells they start undergoing division, what we see is thread-like structures which are called as the chromatin threads, which were observed by Hofmeister in 1848 for the first time. In 1869, DNA was discovered by Friedrich Nietzsche. The chromatin threads observed during cell division were named as chromosome by Waldier in 1888. Friends, chromosome, the word chromosome comes from two words, chroma and som. Chroma means color and soma, yes, som means body. So colored bodies, that is how they were described earlier. But it was Waldier who had named them as chromosomes in 1888. Sutton and Boveri, 1901 to 1903, observed parallel behavior of chromosomes and proposed chromosomal theory of inheritance, that is, chromosomes are the carriers of hereditary characters. Now, friends, if you hear carefully and read carefully, you will get to know that Mendel's theory said It's the genes which are the carriers of hereditary characters. And Sutton and Boveri observed that these are the chromosomes. The basic reason behind it was later on established that there is a similarity in their behavior. The reason was very simple because the genes are located on the chromosomes. Sutton and Boveri suggested that the chromosomes can be equated with the Mendelian unit factors, rather it can be called as the genes that is located on the chromosomes. This concept was further expanded by T. H. Morgan, A. H. Stutillant, and C. Bridges on fruit fly Drosophila. The term factor as the basic unit of inheritance was replaced by Johansen in 1909 with the term gene. So the term gene came into existence in 1909 and it was given by Johansen. So you need to keep this in mind. Now let us observe the historical account in a nutshell. Friends, this particular tabular form is going to be very, very helpful for you for the competitive exams. How can I ask you this? Either I can ask you an individual question on either of it. Yeah, I can ask you the same in the form of match the following, get true or false. This kind of MCQs are likely to be framed from this particular data. So in 1848, Hofmeister observed the threats during cell division. In 1869, Measure identified a cellular component which has high phosphorus and he called it as nuclein. 1888, Waldier observed the threads which were observed by Hofmeister and labeled them as chromosomes. In 1901 to 1903, Sutton and Boveri contributed the chromosomal theory of inheritance after observing the parallel behavior of chromosomes and the Mendelian factors. In 1909, Johansen coined the term gene. In 1923, Bridges coined the term, or rather gave the concept of chromosomal aberrations. In 1928, Frederick Griffith found out that nucleic acid is the basis of inheritance. In 1944, Avery, McLeod, and McCarthy gave the importance of DNA as genetic material in the inheritance of the bacterial cell. In 1948, Lederberg and Tatum did an experiment on transduction, anti-even bacteriophages, and confirmed the nature of genetic material as DNA. In 1952, Hershey and Chase said that DNA is the sole carrier of genetic information from parent to the offspring. So friends, this is very, very important. So keep studying. You can you can note down this and keep on revising this very thoroughly. I hope you like it. Please subscribe if you haven't and hit the bell icon if you haven't yet done it. And please share it with your friends and do comment and suggest if you have to. Thank you so much.